Hi everyone, this video is going to go over the instructions for doing the cancer experiment, Mitosis Gone Bad. Let's start with the background, where we already know that the process for making new identical cells is called mitosis. Organisms do mitosis so that they can grow or replace damaged tissue. And you can see in this diagram right here that there's been some sort of a wound and that cells that are identical to the cells surrounding it need to go in place so that it seals that wound and that tissue is pretty much back to normal. We've learned that before mitosis can happen, DNA has to be replicated so that there is a copy of DNA for each of the new cells. Sometimes during DNA replication, the wrong base is put in place. If spell check enzymes don't catch that mistake, a mutation is left behind. A mutation is an error in the DNA. Most mutations don't hurt an organism. However, too many mutations can cause a cell to die or to become cancerous. You can see the normal cells up at the top are very uniform and regular looking, like the cells that we've been talking about all during this year. But cancerous cells, you'll notice, are all jumbled up, they're all different sizes, and you can just see that they don't look like normal cells. Cancer, the disease, is when mitosis is abnormal, and this causes a cell to divide too fast. As a result, the cells pile on top of each other and create something called a tumor. So let's get to the lab. In this case, we're going to be doing data analysis for this experiment. So read the experiment along with me. A scientist wanted to determine the effect of cancer on the number of cells in mitosis. That would be the number of cells that are reproducing. The scientist compared normal healthy liver cells with normal skin and skin cancer cells. You can tell a cell is doing mitosis because the DNA in cells doing mitosis is condensed or packed up into many chromosomes. A cell that is not doing mitosis has diffuse or spread out DNA. All of the cells were taken from the same person and were allowed to grow for the same amount of time. Now, you can see a picture of what actual cells look like and be able to tell if they are in division or not. These cells right here, these cells, you can see where the nucleus is because it's a dark pink color, but you can't see individual chromosomes. But we have several cells that are actually dividing. In this cell right here, you can see the chromosomes. They're the little stringy-like things that are on the inside. You can also see that this cell right here is going through mitosis. It has chromosomes that are visible as well. The rest of the cells in here all seem to be normal, non-dividing cells, cells that are not in mitosis. So the first thing that you need to do is to stop the video, take a look at the description of the experiment, write down the IV, the DV, and the 3 constants for this experiment. On your lab, you should see 3 cell samples. I'm going to be using the normal liver cells to model for you how to go through the procedure. The procedure is not written on your lab, so you will need to refer to this video to help you to remember what you need to do. So let's read together. In the liver cell sample, count the number of cells not in mitosis and record in the data chart. In the sample, we can see cells that are not dividing look like this. We can see a nucleus as a dark spot in the inside. So we're going to count these. 1, 2, 3, 4. I'm going to ask you to stop the video, continue your counting, and when you are finished, turn the video back on. So how many did you get? I got 46. So I'm going to record 46 where it says cells not in mitosis. Now it says in the liver cell sample, count the number of cells in mitosis and record in the data chart. That would be this cell, this cell, this cell, and this cell. So I have 4 cells that are in mitosis. The procedure tells us to add the cells not in mitosis and the cells in mitosis together. So I'm going to take 46, I'm going to add 4 to it, which is going to give me a total of 50 cells. Now we need to calculate the percentage of cells in mitosis. On our chart, it tells us exactly how to do this, but I'm going to model it here. So we're going to find the cells that are in mitosis. That's this number right here. So for this particular cell, that's going to be 4. I'm going to divide this by the total number of cells, which is this number here. So that's going to be divided by 50. And when I'm done with that, I'm going to multiply it by 100. 4 divided by 50, when I put that in my calculator, ends up being 0.08. And I'm going to multiply that by 100, which means that I am going to get 8. And 8 is the percentage of cells that happen to be in mitosis. Now you know how to follow the procedure, so I'm going to ask you to do the same for the skin cancer cells and for the normal skin cells. Pause the video while you do your data and turn it back on when you're ready to do your graph. Now we're going to graph the data. Our graph is going to need to be a bar graph because our independent variable is actually a category. It's a type of cell. So together, we did the liver cell. So this is where my bar for liver cell is going to be. Your bar for your next cell type will be here, and your next cell type will be here. So you'll have to look at your data chart and put the other 2 categories in place. Over here on the side is going to be our percent of cells in mitosis, because that's what we're being asked to graph. And because this is a percent, it's an easy one to do. So 0% will be here at the bottom, and then 10% and 20%, and you're going to need to put in the remainder of the numbers. So down here at the bottom are cell types, and you are going to need to make sure that you complete your graph. Remember that when we do bar graphs, when we get done, we put the actual data number on top of the bar. Don't forget to put the IV and DV on the title of your graph. There are also questions on this lab that need to be completed. When you're finished, make sure that you submit your completed lab up to the assignment on Schoology. Good luck!